Hey, I just wanted to quickly plug that if you guys want any tutoring, some new slots have opened. If you look in the description, there's a link to my physics and maths tutor profile in which I do tutoring on there.
Hello everybody, welcome back to another episode of the A-Level Cookbook. Today we're going to be doing the OCR A-Level Biology A 2020 Paper 3. A student dissected a kidney. Figure 1.1 shows one half of the dissected kidney. So we can see the different structures that are overall present. So we've got our cortex in the outer part here, right? And then within it we've got our medulla. And then this inner part is your renal pelvis. So it says draw a simple diagram, and then on the diagram label the pelvis, medulla, and cortex. So when we're drawing a simple diagram, it needs to be a continuous line thing, no shading, no cross-hatching, and don't add anything you don't see. So in the exam report, it says that people are adding things like blood vessels and stuff like that. There are no blood vessels in this picture that are obvious, so we're just going to draw what we see. So it would be a kidney-shaped bean like this, and then you would just add the extra layers in. So, and then there. Obviously, and then there. You'd probably draw this a little better than I do, but there you go. And then you would clearly label each component. So cortex, and then here, it's a pelvis, and then this middle-y bit here is the medulla. There you go. So a photomicrograph of a stained section of kidney tissue is shown in figure 1.2, and they've very nicely labeled it for us. So it says state one function of the distal convoluted tubule. So the distal convoluted tubule has got multiple purposes, one of which is that it can have varying amounts of aquaporins in it due to ADH, which means it can be involved in the reabsorption of water. So you could write that. As well as that, if the body's got not enough salt in it and not enough sodium ions and chloride ions, and what you can do is the DCT can actually pump sodium ions back out and reabsorb those, and chloride ions will follow too. So any one of those is fine. I'm just going to go with the reabsorption of water. Now it says describe the function of the structures labeled A in figure 1.2. So if we come back up here, what is A? So we're looking at the proximal convoluted tubule, and this A is pointing at this lining here. Now the point of the proximal convoluted tubule is to reabsorb all of the glucose that's pushed out. So your kidneys, generally how they work is you've got your nephron starting here with your Bowman's capsule. You go proximal. Proximal means nearby. Convoluted means twisty, right? Tubule. And then you've got your descending limb of the loop Henle, then the ascending limb, because it's going down and then up. And you've got your distal, so that's further away. Distal means further away. Convoluted means twisted. Tubule. And then you have your collecting ducts here, right? Now, the proximal convoluted tubule's here, and while we're here, we've got our afferent arteriole, a bundle of capillaries known as the glomerulus, efferent arteriole, and a whole load of capillaries wrapping around the whole thing until you're back to your vein, right? Now, what happens is, because there's a higher pressure in the afferent than the efferent, then water and solutes get shoved out, almost like tissue fluid being formed, into the Bowman's capsule. Once it's in the Bowman's capsule, it travels along the length of this whole thing, and a whole load of reabsorption happens, right? So that first step of everything being shoved out is ultrafiltration, and the reabsorption along the way is selective reabsorption. Now, if we look at the PCT, the entire point of the PCT, like I said before, is to absorb glucose, all of it. And it has specific adaptations to allow it to do that, one of which is that it has microvilli, which means it increases the surface area over which substances are reabsorbed, and they also have lots of mitochondria as well, because what they do is that they use the active transport of sodium as well, similar to how the gut works to reabsorb glucose. So that's another adaptation of them. But one big one is that they have microvilli. Now, since in this diagram it's pointing at this lining here on the inner part of the tube, I'm going to put the microvilli there because that's what it's alluding to. It's not really talking about the cells themselves, so it's not going to be mitochondria. So here, I'm going to write, it has microvilli, and this increases the surface area for reabsorption, right? But as well as that, you could also say that it has co-transporters as well for the absorption of glucose and sodium. So it's very similar to how the gut works. So now it says, which lumen, B or C, has the highest concentration of urea? Explain your answer. So if we have a look at what B and C are, so B is the lumen of the distal convoluted tubule, and C is the proximal convoluted tubule. If we follow the journey of what's happening, so again, we're in the Bowman's capsule, going down the proximal convoluted tubule, reabsorb all our glucose, amino acids, and a good amount of water. It goes down the descending limb, up the ascending limb, right? At the ascending limb, what happens is you shove out sodium ions by active transport along with Cl- following. And what that does is it lowers the potential of the medulla, which is where all of this is happening. As a result, water starts moving out of the descending limb, because that's only where water is permeable, right? The whole point of that first step is to lower the water potential so that it forces water out this way. As a result, when it's moving down here, your water potential is starting to decrease, and here there's barely any, there's a lot less water than there is. Then in the DCT and collecting ducts, you'll have a bit more water reabsorption due to aquaporins and all that kind of jazz. And then, bang, urine. So the question's asking what the concentration of urea is in the proximal convoluted tubule versus the distal. Well, when we get to distal convoluted tubule, a lot of water has been reabsorbed in this stage here. So that means the concentration is higher here. So in that case, it's going to be B, because water has been reabsorbed earlier in the nephron. If you have less water, you have a lower water potential, but all the urea is still in there, so you've got a higher urea concentration. So water reabsorption in the kidney is controlled by endocrine and nervous systems. Aldosterone and ADH are hormones that act on the kidneys. Aldosterone causes sodium ions to be pumped from the collecting duct cells into the tissue fluid. Describe how the endocrine and nervous systems work together to increase water reabsorption from the collecting duct. So we'll talk about the ADH one first, and then we'll talk about aldosterone after. So they're trying to test you to see if you can apply your knowledge of how aldosterone works, because they don't really actively teach very high amounts of detail with how aldosterone works, especially in this curriculum. So if we start by saying we're looking at the endocrine and the nervous systems, and we're working together to increase water reabsorption from the collecting duct, right? So if we have a look at how we increase that, so number one is we use aldosterone, right? As that's one way. And the other thing that we use is ADH, which is anti-diuretic hormone. And the name tells you what it does. Stop, diuretic. Diuresis is peeing. So stop peeing or urinating hormone. So this hormone works by stopping you from peeing. Now, I think a lot of people end up overcomplicating ADH when it shouldn't really be that tricky. Now, don't worry, that's not me being like, oh, you're so silly. What I mean is, the whole point of ADH is to stop you peeing, and what it literally does is it increases the amount of aquaporins in the collecting duct and a bit of the DCT. If you have more aquaporins in your collecting duct, then that means there's more water leaving that and less in the urine. The rest you can kind of work backwards. If you know the overall reason on how this works, then you know the rest, right? So if you increase the amount of aquaporins, more water can leave, less urine, so you're retaining more water. But the question's saying how we use the nervous systems as well, and that's where the hypothalamus comes along. So the secretion of ADH involves using both the endocrine and the nervous system. So what happens is you've got osmoreceptors in the hypothalamus, and what they'll do is they will detect, because they're receptors, remember they detect a stimulus, so they detect a lower water potential in the blood. What they do is they send impulses, because that's the nervous part, right, to the posterior pituitary gland in the brain. The whole point is that then the pituitary gland secretes ADH into the blood. That's your nervous part. The endocrine part is now when the ADH has been released. So what happens, all hormones will be made at one cell and then travel along in the blood wherever, get to somewhere else, bind to receptors, and then do something, right? So in the same sense, ADH travels in the blood to the kidneys. And like many hormones, especially like ADH, it will bind to complementary receptors on cell membranes of the collecting duct and DCT. What then happens is it causes a whole load of stuff inside, right? But the main thing, and then that's because what it does is it triggers the formation. So this triggers the formation of cyclic AMP, right? And then that acts as a second messenger and triggers a cascade of reactions in a cell. So triggers cascade events in a cell. And the main thing that ends up happening is that vesicles containing aquaporins, you write the whole thing, I'm just going to AQPs for short, fuse with the cell membranes, and this increases the permeability to water. As a result, more water is reabsorbed into the blood, and that's an example of negative feedback, right? It hinges on just remembering that here's a cell, right? There's ADH binding to a receptor. Then what happens is these vesicles containing the aquaporins fuse here, and then you end up with a cell with more aquaporins here, so water can leave like that. And as a result, you make less in your urine. That's the ADH bit done. The questions told you about aldosterone, what it does into tissue fluids. We need to look at the aldosterone part and then work out how sodium ions being pumped from the collecting ducts into the tissue fluid increases our water reabsorption. So let's talk about it step by step. Imagine that's the lumen. These are your DCT cells, right? The questions told you that we're pumping out sodium ions into the tissue fluid like this, Na+. We have to exchange them for something, just like with all our other, like the gut, for example. So we're going to have to, exchange some potassium in potassium ions, right? What that does is now the water potential in this fluid is lower, surrounding the cells, the tissue fluid. Now, because we have a concentration gradient, and we've also just lowered the sodium ions in here, sodium ions will move from the lumen into this cell here. As a result, the water potential in the cell then drops, so water is now going to move and follow by osmosis into this cell, and then by proxy into there like that. Because we're shoving the sodium ions out like that, the water will also follow. So that's how it works. We've got to work with what the question's saying. So aldosterone causes Na+ ions to be pumped out of DCT cells into the tissue fluid in exchange for K+. What this does is it lowers the water potential of the tissue fluid and lowers the concentration of Na+ inside the cell. Whenever you're doing these kinds of questions, you should think about the concentrations that are being changed and created, and also the water potentials that are created and changed. Because then it kind of makes sense. Because then as a result, the sodium's going to want to move in there, sodium ion size. So then Na plus enters the cell via diffusion, or facilitated diffusion really, and this lowers the water potential of the cell. And what happens is water enters the cell from the lumen via osmosis and then into the tissue fluid. And then that's reabsorbed. So that's the other hormonal element of it. There you go. So diuretics are drugs that decrease the reabsorption of water into the blood and from the kidney. They can change the concentration of ions and other molecules in the blood. Some diuretics are used to treat high blood pressure, and here's 3 different diuretics, X, Y, and Z, some of their effects in the body. So the rate of urine production without is 1. Without a diuretic, the blood chloride ion concentration is this, potassium ion is this, and then glucose is that. With a diuretic, the rate of urine production goes up with X, and this chloride goes down, potassium ions go up, and the glucose stays the same. With Y, the rate of urine production is higher, the chloride is quite higher, the potassium ions are a bit lower, the glucose is raised, and Z, rate of urine production is higher as well, chloride is much higher, potassium ions are higher as well, and the glucose is also higher. Fine. It says suggest which of the diuretics X, Y, Z would be most effective at reducing a person's blood pressure and explain your choice. So if we want to reduce blood pressure, remember, blood pressure means that there's too much going on, like speed-wise, right? It's way too much pressure. So for example, if I have a water bottle and I squeeze it, the water comes out this way because it decreased the volume here. Now, these arteries can't really just make themselves even bigger to accommodate for that blood pressure. What we need to do is, because there's so much happening there, we need to try and reduce the volume of the blood in there. As in, not the volume of the vessel, the volume as in the amount of blood flowing, right? If I have a nice big box here with 1, 2, 3, 4 molecules chilling, if I then make the box teeny, 1, 2, 3, 4, can you see how cramped they are? The pressure's really high. So my solutions to reducing the pressure is 1, increase the volume of the container, like we have here, or 2, I decrease the volume of the materials inside it, as in the number of particles here. We can't really just make the volume of an artery just bigger for shits and giggles. Like, I know arterioles can constrict and stuff and redirect blood, but these arteries aren't going to be able to just get bigger to compensate. So the only solution is to take out some of the blood in there, or the volume of blood, the fluid in there. So the best diuretic in this context would be Y, because it makes you pee the most. If you're peeing the most, you're losing more water, and that means you're going to reduce the blood pressure even more. So then diuretic Y, because it would reduce blood volume the most, due to it having the most urine production. There you go. So then it says, which of the diuretics X, Y, Z would be most appropriate for use by a person with type 2 diabetes, and explain your choice. So we want them to pee more, that's the whole point, but type 2 diabetes is a problem where you have too much blood glucose because your insulin receptors are less responsive to insulin, right? So if we have a look, we need to make sure that they're actually, peeing, right? So it's going to be one of X, Y, and Z, but we want to make sure that the blood glucose isn't getting fumbled around with. Now, if we look at Y and Z, they increase your blood glucose, which you kind of don't want in somebody who's got type 2 diabetes. So the only option here is X, because that stays the same, albeit that it makes them pee less. So here, it would be X, and why? It's because it does not raise blood glucose concentration. There you go. So valves control the flow of blood through the heart. Complete the table to show the roles of the 2 valves in the heart. So the heart is another thing that I find a lot of people struggle with. All you've got to remember is that right side is from like all the veins of your body, right? So the vena cava, and then to the lungs, and the left ventricle is lungs to heart, and then out to the rest of the body. So what I mean by that is if we draw like our right atrium, our right ventricle, left atrium, left ventricle, right? If you just think about the purposes of the ventricles, and it gets kind of easy. So the right ventricle, the entire point of it is to get blood all the way to the lungs. But that's the entire point. All right? And the entire point of the left ventricle, this is a really not accurate drawing, by the way. I'm just drawing it as a schematic. This is not what the heart looks like. The entire point of the left ventricle is to get it to the rest of the body via the aorta. The entire point of the atria are to just get venous blood back in. That's all, from wherever they've come from. So if you think about it in logical steps, if the right ventricle wants to shove blood to the lungs, then that means all of the other venous deoxygenated blood we don't want comes into the right atrium. So that means we have our vena cava here, or inferior and superior vena cava, right? The right atrium will shove blood into the ventricle, and then that'll shove blood to the lungs. So now in the left atrium, if you want to shove all the blood in the left ventricle, right, to the rest of the body in the aorta, we want to make sure we have nice, beautiful, juicy, oxygenated blood. So then what happens is we have things from the lungs coming back into the left atrium. Now, veins, V in, takes blood back into the heart. Arteries are away. It's nothing to do whether they're oxygenated or not, or deoxygenated. A lot of people like to say, oh, well, veins are deoxygenated. Well, that's objectively wrong, because the pulmonary vein, the vein from the lungs, is oxygenated blood. So it's the vein that's in an artery that's away that's key. So into heart, and then A is away from heart. That's it. Now, obviously, there's a bit more to this. It's a bit juicier than that. But if you just know the general directions of which everything's going, then it gets a little easier. Again, right ventricle, the entire point is to get blood to the lungs. Left ventricle, the entire point is to get it to the aorta. These are both connected to arteries away. These are both connected to veins in. Now, obviously, there's a bit more that your heart does. Number one, the other thing I see is that these two contract at the same time, then there's a pause, and then these two contract at the same time. It's not this, then this, then this, then this. They're both at the same time because you want the blood to go this way and this way. Otherwise, if you did it one at a time, your heart would kind of bend like that and you'd like die. And it's just not compatible with life. So again, there's that. But also to stop the regurgitation of blood back up here, because we don't want it to do that. We want it to go in, out, like that, in both ways. In, out. We have valves. So there's some valves here. These are your AV valves, atrioventricular, it's in the name, or tricuspid and bicuspid or mitral on the left, right? As well as that, we have valves in these arteries as well near the start of them, and that's your, if it's the lungs, pulmonary valve, or you can call it semilunar, and then there's another valve here, which is another semilunar. And the valves will only open, right, if the pressure behind them is high enough. So the pressure behind them is high enough, it will open like that. Whereas if the pressure's too high in front of them, as in, like, you know, for example, the ventricle's not contracted hard enough yet, they won't open. That's the key thing to do with all of the heart in a nutshell. Obviously, there's all the other bits and bobs about it, but that's pretty much the big one. So if you have a look, it says source of blood, valve that controls blood flow is the right semilunar valve, the destination of blood. So semilunar valves are in your big arteries, right? If it's on the right side, like I said before, the whole point of the ventricle is to put blood into the lungs. So if that's the case, then it's going to be to the lungs, and it's an artery because it's away from the heart. So the fancy word for lungs is pulmonary. That literally just means lung and artery. There you go. Now the source of blood then would be where it's going, straight into this right semilunar valve. It's not the right atrium, it's going to be the right ventricle because that's what's contracting and then shoving blood into that, like I said before. So right ventricle, right? Cool. So then it says left atrium. What is the valve that controls its blood flow? So the left atrium drains into the left ventricle, right? So it's going to be the left AV, or you could call it bicuspid, or you could call it mitral valve. And where does it go? Left ventricle to shove out to the rest of the body. There you go. So ventricular septal defect is a hole in the septum of the heart. The diagram shows the heart with VSD. So we've got our ventricular septal defect. That should be just fixed out like that. Shouldn't be a gap, right? Fine. So now it says, describe and explain why people with VSD can become easily tired. So here, the problem is now we have a hole in between the two ventricles. Generally speaking, any fluid will, just anything in the world actually, likes to go from a high to a low pressure, right? If I squeeze a bottle, the water, and the lid is open, the water comes out because the pressure now in the atmosphere is way lower than inside the bottle. In the same sense, because now we have a hole in the heart, we're going to start having things moving around because of changes in pressure. Your right side of the heart generally has a lower pressure because the right ventricle is a lot less thick, because it only needs to get the blood to the lungs. Whereas the left ventricle is fatter and chunkier and it's got to get a stronger contraction going. As a result, what happens is, so normally again, right atrium drains blood into the right ventricle, and then the right ventricle will shove it into the pulmonary arteries, right? Then in your left atrium, it will drain with your pulmonary vein and all that jazz. And then what will happen is your left ventricle contracts and it shoves it out into the aorta. Now the problem is that we have this gap here now. Now fluids and everything in the universe likes to take the path of the least pressure, right? So if the contraction here in this ventricle is quite strong and this has got a low pressure, what will happen is that oxygenated blood will enter the right ventricle like this. As a result, this left ventricle blood is now in the right ventricle and it will go to the lungs. Because now in the lungs, you've got more oxygenated blood getting there. You've got a less steep concentration gradient, so less oxygen's actually being reabsorbed, right? The rate of diffusion of oxygen back into it is quite slow. So then when this blood returns to the left atrium and then gets shoved out again, you've now got less oxygenated blood getting around the body. And as a result, you've got less oxygen available for aerobic respiration. And then boom, you are now tired. So it takes a couple layers of thinking this one. So here, what happens is that you get mixing of left ventricular oxygenated and right ventricular deoxygenated blood. And then as a result, more oxygenated blood gets to lungs, less O2 is absorbed. So then there's less O2, O2 blood, oxygenated blood really, traveling around body to cells. I should say pumped really, that's a better word. Whoops. Pumped. Therefore, there's less O2 available for aerobic respiration. So then there's less ATP and less energy. There you go. So can you see how people easily become tired, to me, is hinting at something to do with energy. And it's a problem with the heart, which is all about getting blood in the right directions and stuff. So it's every single step all the way until you can get to explaining how they become tired. Otherwise, you'll miss out points and not get the marks. So creatine kinase is an enzyme that catalyzes reaction in heart muscles. High level of creatine kinase in the blood indicates that the person may have had a heart attack. So just why high levels of creatine kinase in the blood indicates that they may have had a heart attack? Well, it's told you in the question that this is an enzyme that catalyzes reactions inside heart muscle. It shouldn't be in the blood. It's inside the muscle. So the idea here is that this is an enzyme that's found in cells, as in the CK is intracellular, or you can say intracellular, or it's an enzyme only found in cells. Therefore, if it's in the blood, it means that its tissues must have been damaged. It's released into the blood when tissue damage occurs. And this is something we can measure in people with heart attacks. It's CKMB, or another one we measure is troponin as well, cardiac troponin. So mice are often used in laboratory studies to research treatment on heart conditions. These mice are often clones. So just one reason why clones are used in this study, right? If you're thinking about it this way, your independent variable is the treatment. The dependent variable is their response to treatment. I know that seems a bit pedantic, but then if you think about it as, what could screw up what I'm measuring? The treatment responses. What can screw up the dependent variable? Now you're like, okay, right. Something to do with clones is stopping their responses to treatment being screwed up. And would you look at that? That is indeed the answer, is that it ensures the responses to treatment are the same. Responses to treatment are the same. A perfect experiment in this sense, when you're using lots of mice, is you want to get the exact same response and you want it to be repeatable. And then also reproducible, which means you've got a very reliable, good, valid experiment that can actually hold some weight. So now it says students investigated the effect of light on a garden of cress seedlings. A total of 120 seedlings are divided into two groups of 60. Group A was grown in darkness for 2 days. Group B was grown for 1 day in darkness and then for 1 day in white light using the setup shown in this figure. Fine. Let's have a look. So then they say they've got the seedlings, they've got water, they've got white light going this way, and it's transparent glass. And then here we go. It says the results of the students are recorded in this table. So we've got mean length of the stem and the root, mean mass of the stem and the root, and it's group A and so forth, right? So the independent variable is like dark versus dark plus light, right? And then our dependent is the stem, root, mean, length, or mass. This is useful because it helps, again, just pinpoint where things need to be controlled or this or that, right? Fine. If we have a look then, so remember that group A is just pure darkness, whereas these geezers were dark plus light. So if you have a look, the mean length of the stem was 13, but the root was 18, whereas if you then compare the stem in darkness versus dark and light, the stem is longer, the root is also longer. Fine. And mean mass, the mass of the stem is 102. When you've got in darkness, but with dark and light, it's gone up. Whereas in dark and light, the root mass was 60 in the dark, but in dark and light, it's 120. So compare one by one and then with each other like this, right? So if we have a look, the difference between these two was not too high. And then in dark and light, the difference between these two, again, is not too high. Whereas if we have just darkness for the mean masses, the difference is pretty heavy, right? The stem is has more mass, heavier than the root. And over here as well, darkness plus light, the difference between these two is also still roughly the same heaviness. Fine. As in there's, about 40-ish between them. But it always helps to do one by one, like this, one by one, one by one, one by one, and then side by side, side by side, side by side, side by side. Because it always just helps figure out, what patterns you might spot. Now here as well, we've got another table, and it says direction of growth in group B, away from light, stem, and root, neither towards or away, that, and then towards light. And this is the number of seedlings, right? So we have a look. Away from the light was only 2, but a lot of roots went for those, and neither towards or away was also just a few of the stem, a lot of the roots, and then towards light, lots of stem, barely any roots. That kind of keeps in with what we want to expect, right? Now it says describe and explain the results shown in tables 3.1 and 3.2. Remember that this question was about putting seedlings in, right? So these are the seedlings. They're chilling, and then here we've got a length of a stem and a root, right? So if we have a look again, when we've got light, you can see that the mean mass of the stem and the root is higher, and also the length of the stem and the root are also higher, whereas in darkness it's not as high. So something's different about how these seedlings are behaving when you have light. It must be, any bells ringing here? For me, I'm thinking germination of seeds, because you've got light that might trigger the germination of these seedlings, because you can see that they've now got bigger stem and root. So, and that does make sense because if you think about it, they are important in the elongation of plant stems during the growth. And without gibberellins, you find that a lot of plant stems tend to be shorter as well when you've not got gibberellins present versus when you do. So that clearly is an element of, what might be going on here. The reason I'm going for gibberellins is because they're involved in germination and they're produced in seeds. They promote seed germination and also, stem elongation and flowering. So here then, describe and explain says, say what you see and then explain why, or come up with a reason why, right? So in B, light led to an increase in the mass of stems and roots. Therefore, it must be that there's more growth, right? So now I've done my describe part. I like to do it bit by bit, right? So I'm explaining the first finding, and that's the key first finding. Now I'm going to explain what might be going on. So a reason that this could happen is that light might trigger, because these are seedlings, remember? Light may trigger germination, and this will this could increase, the rate of photosynthesis, because we're talking about them growing, right? And the way they're going to grow is eventually by having photosynthesis happen, which means more carbohydrates and more biomass, right? Cool. So that's that first one explained. Are there any other results that are, very key between comparing these two? Not massively, because I was saying, as comparing side by side, the differences between the two aren't very big compared, this way and this way. Fine. So then the next table to look at is if we look at the table 3.2. So table 3.2 is showing the direction of growth, right? With this table, what I'm going to do is I can say that if we have a look, most of the stems grew towards the light, very few are away, and very few with neither, right? Whereas if we have a look here, nearly half, because if you work that's like 33 versus 29, nearly half of them grew away from the light. A lot of them were indifferent, and very few went towards. So that's what we're going to say. That's our describe part there. So most stems grew towards light, around half roots grew away, and a few did not change direction, right? And then we need to think about why. So what's causing cells to bend is cough, 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 auxins, woohoo. If I'm looking at things bending towards light and away, what I'm looking at is cough, 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 auxins, especially indole acetic acid. So if we have a stem or a shoot, what happens, right, is if I shine light, for example, this way, remember that in indole acetic acid has different effects in shoots versus roots, right? What happens is in shoots, it will promote elongation of cells. So if we want this shoot to bend towards the light, it will bend like this, but there'll be more auxin on this side, more IAA on this side, because then this will promote the cells to elongate, but these won't elongate, causing it to bend towards that light. Whereas in roots, if I have a root here, for example, and I've got light here, then in roots, it has the opposite effect. Indole acetic acid actually inhibits elongation. So what it'll do is it'll bend away from the light downwards, but there's more indole acetic acid here, and it inhibits it. So these will continue elongating normally, because that's what they'll do. Cells just grow, right? But this will be less growy, so it bends them down like that. So in summary, in shoots, stems, things like that, indole acetic acid will move to the shaded part, and then it promotes elongation, right? Making them bend that way. Whereas in roots, it still goes to the shaded part, but it inhibits it instead. The effects are the opposite. So to explain this then, we're just going to say this could be because of indole acetic acid or auxins. So what can happen is that IAA, where you write the whole thing, right, moves to the shaded side of the stem and increases in concentration, and this promotes elongation of cells, and the stem bends towards the light. And like I said before, the opposite effect is seen in the roots. So in the roots, you still get it still, accumulates. So IAA still accumulates on the shaded side, but instead will inhibit cell elongation. Therefore, it will bend away from the light. But that's not it. We have to describe all of the results here, right? You can't just leave out the things that don't look exciting. We have to figure out why on earth a decent chunk of them were neither away or towards the light, right? Because we're not really expecting that to happen. So what we need to do is we've got to go and have a look at possible problems in this experiment. As in normal circumstances, it should be, again, it should just be towards or away. So why on earth are we getting some weird stuff going on here, right? If we have a look, we've got this white light going here. Now, immediately, one thing I'm thinking of is that maybe it's not really, we don't just have pure white light in natural environments. It's kind of, a bit of both, a bit, sky's blue, all that kind of jazz, right? A bit of these wavelengths and that and whatnot. So it might be that just because it's run in an artificial setting, we're not going to get the exact same results, is what I'm thinking. So some roots were towards and some indifferent, not indifferent, but some didn't move at all. You've got to explain why those funky results came along as well. Indirection, sorry. Could be due to non-natural light or experimental setup. As well as that, you could talk about the fact there's no soil and things like that, right? And the conditions aren't there. Or even some of the other things it says as well in the mark scheme is stems are heavier than roots, so tips, and also shading of stems. But I think that's probably the biggest one. It's, in these kinds of experiment questions, always talk about the obvious funky, cool stuff. We're yay, woohoo, this did happen. But that is a pretty high number of them to not do anything, and that's a bit weird. So we've got to explain the weird result as well. You can't just leave that out in an experiment. So now it says the students wanted to test whether there was a significant difference between the stem lengths of the seedlings in A and group B. What's the most appropriate statistical test? They're looking at the mean length between A and B. They want to see if there's a significant difference in the means between A and B. This is going to be an unpaired t-test, because the whole point of an unpaired t-test is you're comparing the differences in the means of two groups. For example, the height of one group versus the other, and the means, to see if there's any significant difference in their means. When you're talking about p-values and probabilities and all that jazz, you're looking at that stats test, you need to identify what the p-value is testing. So in a student t-test, or an unpaired t-test, the p-value is testing to see if there is a significant difference in the means of two results. If you're doing correlation coefficient, the p-value is testing to see if the correlation is significant. In a chi-squared test, we're looking to see if the p-value is testing whether the difference in expected and observed is significant. You really need to knuckle down what that p-value is testing when you're answering your stuff. Now, the reason this is unpaired is because we have two separate groups. A paired test would be where you have like one singular group of something, and then you're looking at the means between the same group after something's happened. So it could be like, I don't know, a group of students doing a run, and you're working out their mean pulse, and then you like work it out at rest, and then after the run. It's the same group, so it's paired data. So you would do a paired t-test. Here it is unpaired because they're two separate groups. They're not the same group undergoing like an intervention and then before and after kind of thing. So it says justify your choice of statistical test. Oh, it's exactly what I just said there. It's because we are comparing two means. There you go. So table 3.2 records the direction of growth as away from light, either away from light, or towards light. And then it says the students used the chi-squared test to determine whether the direction of root growth was significantly different from their expectations, and their null hypothesis was there is no difference between the expected direction of root growth and the observed direction of root growth. The students compared their chi-squared value of 8.10 to the table here. Then it says what can the students conclude about their results based on this chi-squared value? So here we need to figure out the number of degrees of freedom, right? Well, how many classes do we have? So we've got 1, 2, 3. We have 3 different possible phenotypes or outcomes or things, right? The number of degrees of freedoms is your classes minus 1. So it's going to be 3 minus 1, which is 2. We want a probability, a p-value of 0.05. So we're looking at 2 and 0.05 here. So we're looking at this. If your value is greater than this critical value, right, it means that you've met that 0.05 threshold, okay? And we have. So then we can start by saying that. We'll start by saying that the chi-squared value of 8.10 is greater than, and you need to make sure you make it clear, 5.99, right? Therefore, the probability of, and then we need to figure out what it is that they're saying. What's the p-value testing? The p-value is testing the difference between the expected and the observed direction. So you will say that, or just say the results. That's not helpful. The probability of the difference in expected, whatever it was, versus observed, I'm just going to write E versus O for short. You obviously write the whole thing. The probability of the difference in E versus O being due to chance is less than 0.05 or 5%. Therefore, the null hypothesis can be rejected as the difference is significant. This difference we just established. Because their null hypothesis said there is no difference between the expected and observed, but we've just done a chi-squared test and the value says there is a difference, so hence you can reject it there at that p-level, at p-value. The solutions in which the seedlings are grown contain various dissolved ions. The table shows 2 of the functions of these ions, and the seedlings complete the table with the formula of each one. So it says it supplies elements that form parts of the structure of the amino acids and nucleotides. So in that one, it's nitrates, NO3 minus. Although it says allow ammonium as well, that's 1. Then it says forms part of the structure of DNA and phospholipids. So DNA has a phosphate, ribose, and nitrogenous base, and phospholipids, phosphate, and the lipidy bit, right? So then in that case, what's common to both of them is phosphate, which has the formula PO43 minus. The students also investigated the effect of plant hormone concentration on root growth. State the name of a plant hormone that would be expected to affect root growth. So then it's going to be IAA. Obviously, write the whole thing. I'm just lazy. I'm just going to write IAA. Now, in this investigation, the students controlled light, temperature, and mineral concentration. State one other factor they should have controlled in this. So remember, we're looking at how much is screwing up the bending of things, right? And we need to think, what could screw up the bending and the growth of these things? I mean, there's a lot of factors that come into it. 1, you could say the pH. Number 2, you could say the species of the plant, the age of the plant, the size of the plant, how much water there is. You could also say how the seeds would, like pre-treatment of seeds, what was actually done with the seeds, where were they from? Lots of them. It's just anything that screws up the dependent variable. So the growth of plant roots is thought to be controlled by specialized cells called statocytes. One hypothesis for how a statocyte controls root growth involves small organelles called amyloplasts and is shown in this figure. So you've got the position of root, gravity is down, whatever, and here as well as that. And it says falling amyloplast seems to be linked to the gravity here. And then what happens is you've got our endoplasmic reticulum. You can see the CA2 plus moving out, and then it causes the direction of growth going down. Whereas if we have gravity this way, we've got the falling of the amyloplasts as well into this what looks like the endoplasmic reticulum. Again, we've got that CA2 plus coming down, and the growth goes down. So then it says, what can you conclude about how a statocyte controls root growth in this figure? So what must be going on is that they're falling, they smack into this thing, and then all of a sudden CA2 plus comes out in the same direction as gravity, right? So first of all, I'm going to write that these amyloplasts, because it's the same direction as gravity, fall with gravity. And obviously, if they're doing that, then they're interacting with the ER. So like they come into contact with ER, and this leads to CA2 plus release in the same direction as them falling, because that's what you can see here, right? And then, for some reason, it causes growth in these directions. So clearly, this CA2 plus, according to this diagram that is, stimulates growth factor release, GF for short, promoting cell elongation or growth, because that's what the diagram is showing. You know, it's showing that these are falling, and all of a sudden, bang, they hit this, and then calcium comes out, and then we get growth, right? So if that's happening, something about this calcium is causing elongation or growth. There you go. So prokaryotic cells have cytoskeletons. The molecules in these prokaryotic cytoskeletons are different from the molecules in eukaryotic cytoskeletons, and there's 3 molecules present in a prokaryotic cytoskeleton in this figure. Protein A, which is here, so we've got a ring of this contracted protein A. Protein B is similar in structure to actin, and then protein C is we've got this bacterium there just chilling with protein C. So it says suggest the function of protein A. Well, if we have a look here, can you see how it's like there's a ring of it there, right? And it's contracting, and then all of a sudden we've got 2 cells after, although we've got some weird mysterious protein B in the middle that's not been shown in a diagram. If for some reason we've gone from one to next and there's a contraction going on and now we have 2 cells, for me that's going to be the cell membrane splitting. So in this case, cytokinesis is probably what the role is, or binary fission's allowed as well. So state the function of protein C. So if you have a look at protein C, what's going on here is after that, we remember we've got a new cell wall being made for each of these. So it's something to probably do with helping it have its cell wall until it's being built, or the cell membrane, as in it's maintaining the cell shape or structure. So provides structural support to cell shape. Provides structural support for cell shape. An antibiotic called A22 binds irreversibly to protein B. Despite this antibiotic properties, A22 is not used in humans. Suggests why they've advised it should not be used in humans. Protein B is similar in structure to actin, and it's told you here that it binds irreversibly to protein B. If it binds to actin, that's probably not a good idea. So I mean, one, it would binds to actin, and as a result, it could cause things like paralysis. Because that's what actin's needed for. It's in your muscle paralysis. Probably not a wise idea. Vancomycin is an antibiotic that's been used to treat bacterial infections for many decades. Several strains of bacteria have evolved resistance to vancomycin. Oritavancin is an antibiotic with a similar structure to vancomycin, and here's data from treatments with these two. Independent variable is the antibiotic used, and the dependent is the percentage of patients that are developing these side effects, and also the percentage of bacterial infections that have been cured. So if you have a look at oritavancin, this has been used for 4 years, that's been used for 60 years. And if you have a look, more have nausea, less have headaches. In vancomycin, that is, less diarrhea here, more slight equal vomiting, kind of similar constipation, a little bit more here. These are equal dizziness. Here, this treats Staph aureus a little bit more. This treats MRSA a little bit more. And Streptococcus is treated best with vancomycin here as well. So it says, use the table in 4.2 to evaluate the advantages and disadvantages of using oritavancin rather than vancomycin. So advantages, one by one, we'll go through them. So if we have a look, it's more effective against MRSA, isn't it? And that's the one that we're big and scared about. So more effective against MRSA. And also, there's a lower, there was some side effects that were low. Which ones were they? Nausea is less, constipation was less slightly, and lower risk of nausea and constipation. As well as that, it's been used for fewer years, so there's less of a risk of antibiotic resistance developing simply because it just hasn't been used for as long. But I'll go with those for now. As well as that, some disadvantages. Well, in the same vein, because it's only been used for 4 years, its effects, or its effectiveness is not as well studied. As well as that, it's less effective by a lot with the strep. So it's less effective against Streptococcus species as well, and that's pretty not good. And as well as that, on the flip side, it's got other side effects that are higher as well, but those are the ones I'm going for. Bacteria such as E. coli can be genetically engineered to use in medical science. An example is the genetic engineering of E. coli, as shown in this diagram below. So the gene for beta-galactosidase is inserted into plasmid using some mysterious enzyme. The gene for beta-galactose is extracted using some enzymes. And then here, the rate at which the bacteria are taken up is increased by using something. Fine. So if we have a look then, it says complete the diagram by writing the missing words and then answer on the diagram. So if we're having a look here, so for X, we are extracting, we're taking out a specific gene. We are chopping it out. The way you chop out genes is by using restriction endonucleases. We're getting a fragment out. That's the entire point of them. What they'll do is if you use a restriction endonuclease enzyme or restriction enzyme, it breaks the phosphodiester bonds here, but it also leaves some sticky ends. As a result, if you use the same restriction endonuclease on the thing that you want to modify, i.e. this plasmid, it will form hydrogen bonds like this with its sticky ends like this. And although maybe not technically correct, but it kind of helps me remember is now that it's floating here, it's not actually stuck onto the plasmid yet. The only way to actually get it to stick onto the plasmid is to form new phosphodiester bonds between this part here and here like that, because this is only going to help it line up, right? So it's almost like, imagine if you've got your DNA replication happening, right? And you've got your template strand and these are just lined up. They're not actually joined together to make a whole strand yet. They're just kind of floating here because they're complementary base pairs. What we need to do is we use DNA polymerase to make new phosphodiester bonds between them like this. In the same sense here, this is just going to be floating awkwardly here like that. And it's not actually stuck together to these bases here. So what we end up doing is we use an enzyme called DNA ligase, and that will make new phosphodiester bonds between these to actually stick it in there. Ligase stick together. So then it says the rate at which bacteria take up plasmids is increased by using something. So we need to figure out a way to get them to take up more of these. What you can do is multiple techniques. We can use something called electroporation or heat shock or calcium salts as well. Either one of them is fine. I'm just going to go with electroporation. Use electricity to get pores, as in make holes in them. So it says, suggest why the scientists used a plasmid that contained an antibiotic resistance gene, right? So the whole point is we have taken them up, whatever, blah, blah, blah. And then it says E. coli that have taken up this plasmid are injected into mice with liver tumors. Now, we need to actually figure out which ones have been taken them up. Otherwise, we're just putting non-transgenic organisms into these mice, which is a bit useless. So the idea is what they can do is they can grow in a petri dish, and normally we can use UV light, but what they can instead do is just use an antibiotic, and those that are resistant will survive, and that'll be the one that took it up. So the idea here is that it's used as a marker gene. So it's used as a marker gene to identify which bacteria took up the plasmid. Because remember that the plasmid is what's being taken up. Now they have an antibiotic resistance gene. You need to think, why would they want that in the plasmid? And there you go. The only reason is so you can identify which ones actually took up the plasmid. Because if it didn't, it'd die. So the scientists may observe the following. 1 in 4 bacteria took up the plasmid. 1 in 1,000 of these plasmids taken up contain this gene. So calculate the percentage of bacteria that contained the beta-galactosidase gene, right? So if we have a look, 1 in 400 took up the plasmid. So if I say that X is the original, that's going to be X in 400, right? That took up the plasmid. Took up plasmid. Then it says 1 in 1,000 of the plasmids taken up by the bacteria contain this gene. So out of these 400, 1 in 1,000 of those will also have taken up that gene, right? So now you end up getting X over 400 times 1 in 1,000. So you get X over 400,000, right? Now, a percentage is going to be your value of the original times 100, right? So we end up getting X over 400,000 divided by the original, which was X, our starting thing. So we end up getting 1 over, then times 100. These cancel out, right? Because that would have just gone on the bottom here. So then you end up getting 1 over 400,000 times 100. If we do that, we get 2.5 times 10 to the minus 4, where X is just my original value. We don't know what it was, but it doesn't matter because it will cancel out like that. So 2.5 times 10 to the minus 4. If in doubt, just work through it in algebraic form, and then you may get to a point like that where you're like, oh yeah, there you go, done. So a technique called quantitative PCR is used to check that the E. coli population is growing on the mice liver tumors rather than on healthy tissue. Because remember, in the stem, they've told you that we're injecting these E. coli with the recombinant plasmid into the mice with liver tumors. So it says, suggest how these scientists can use PCR to compare the growth rates on cancerous liver tissue and healthy tissue. So let's just follow the steps so far. So the transgenic E. coli it says here that they've been injected into the liver tumors, right? So injected into liver tumors. And it says here that they're using quantitative PCR to check that they are growing on the mice liver tumors instead of healthy tissue, right? So these are going to be growing on liver tumor. And for some reason, we can use quantitative PCR. When you're growing these on the liver tumor, they're going to be replicating. They're going to be having, DNA replication, aren't they? And PCR also does DNA replication. On healthy tissue, it's told you the question that they shouldn't really be there. For example, if we have 32 of these transgenic bacteria on the liver tumor, right? That's going to be 32 DNA strands, right? And every time you do PCR, it's going to be times 2. Imagine there's going to be less on the healthy tissue. You'll start off at 2, for example, times 2. Can you see how that's gone up to 4? That's gone up to 64. And then if you double it again, 1, 2, 8, that's going to be 8 and so forth. So we can compare the number of DNA copies as we run more and more cycles of PCR between the two of them. Because if there's more on the liver tumor, there'll be more to begin with and more at the end, if that makes sense. So we actually have to talk about the steps, though, because we've only just got tissue. So what we need to do first is extract the DNA from liver and healthy tissue. Then you would, compare. So you'd run PCR on each and, run a set number of cycles and compare how much DNA is present in each. Because if they're the same at the start, if there was 2 and 2, you would expect eventually after 1 and 2 cycles, you would expect there to be 8, right? Whereas if there's 32 at the start, can you see at the end, there's 128 versus 8, so there must have been more to begin with in the liver, so there must have been more growth, if that makes sense. Some people think that genetic engineering of certain organisms is unethical. However, there's very few concerns about the genetic engineering of bacteria, such as E. coli. So just why there are very few ethical concerns with E. coli? Well, they're not animals, so there's not really any pain or suffering in the same sense. So there's fewer animal rights concerns because they're not going to be oh my god, this hurts, ow, we're no, you're making me make insulin, oh please. They're not conscious, they don't feel pain, they don't have emotions, all that kind of stuff. There you go. So ecologists were studying an area that contained 3 different habitats. The area is shown here. So we've got peat, bog, farmland, and grassland. Fine. So the ecologists sampled the area to estimate index biodiversity. Describe how the ecologist should choose the number and locations of their samples to ensure that the sampling is representative. Representative means, for example, if I get a bunch of lactose intolerant people and I have 3 of them and I give them milk and none of them shit themselves, then I can't just go ahead and say, well, they don't shit themselves because there's 3 people in my study and 100% of them didn't shit themselves because it's not representative. The most of the population who are lactose intolerant will probably have diarrhea and upset tummies. So that's not representative. But if I do 10,000 people and a good chunk of them, about 70% of them actually do end up getting diarrhea and stuff, then that's a more representative sample. It represents what real life's more like, right? So we need to decide what kind of sampling we're going to do in this area to investigate the insect biodiversity. So there's options. We can't just throw random intervals everywhere. We actually have to go within these areas and sort of do some sampling within them randomly, right? We can't just do it across the whole thing. We might get 1, 2, 3 here, 1 there, and then 1 there. That's a bit rubbish. So what we do is there's different things. So for example, the way of sampling can be entirely random, which we're trying not to do because again of what I just said. You can have systematic. So you know how you do your transect, right? Imagine there's seawater here and then there's a whole forest there. Systematic means you're doing it in regular intervals at fixed set gaps. So you start here, you do a quadrat, and then 5 meters, quadrat, 5 meters, 5 meters, 5 meters, 5 meters. And that's really useful to see if there's a pattern or a trend in the types of organisms and species and stuff as you're moving from one place to another. That's systematic, right? The next type is opportunistic. So this one is a bit different because this is where the samples are chosen by the person doing the investigation. This is a lot simpler, but the problem is that you get very biased data. And then you've got stratified. So what you do with stratified is where you identify different areas and then sample them separately, and then you sort of scale up the entire thing, right, accordingly. What I mean by that is you divvy up something, for example, here, we're going to divvy up this area, these 3 areas, right? And then what you're going to do is you're going to divide them into something called strata, those subgroups, and then you take random samples from these in proportion to the size of them, if that makes sense. So what I mean here is we're going to divvy up this place here into 3 things, and then we're going to you're going to take out samples in proportion to their sizes of the whole thing. So the entire area is going to be 800 plus 800 plus 400 meters squared, which is 2,000 meters squared, right? This 800 over 2,000 is 40%, and then 40% for the farmland, and then 20% here. So the samples that we would take are going to be proportional to each one of these. So for example, if I decide, okay, I'm going to take 40 samples here, I do 40 here, and then 20 here, right? Or I would do 5 here, and then 10 here, and then 10 here, that kind of thing. So that's all it is here then, the calculation is you're going to do stratified sampling, and then you would take a random sample, or take random samples, sorry, in proportion of 3 areas to the whole, which are, so 20% for the, what was it, grassland, and 40% for the farmland, and 40% for the peat bog. There you go. 2 of the insect species that were sampled were the large heath butterfly and the bog hoverfly. The ecologists used the capture remark recapture technique to estimate the population sizes in 2 different calculations. If anyone, so I've been watching Clone High recently, and if anyone can explain why JFK sounds like error, I am JFK, that would be helpful, because I see that bit everywhere and I don't understand what it is. Off topic, but like anyone can comment why, that'd be very good, because I don't get the joke. It's everywhere, dude. The Lincoln estimate. But anyway, yeah, the reason I say that is because Lincoln reminds me, whatever. Anyway, calculate the population size of the 2 insect species using each of these formulae below, and then write your answers in the table. So we have the Lincoln estimate formula here, and the Chapman estimate formula there. So little n is the number of individuals in a particular sample, and then m is the number of marked individuals. Fine. So all we got to do is just calculate using these formulae. So here, it's going to be n1 times n2 over m. So the total, remember, is the number of individuals in a sample, which is going to be 77 times 73 over 4. So if I do that, we get 1405, whoops, because you can't really have like a fraction of something. And then here in this other one, it's going to be n1 plus 1. So it's going to be, 78 times 74 divided by 5 minus 1, which is 1153. And then for the bog hoverfly, it's the same thing. You just stick them in and you get 30 and 20. The Lincoln and Chapman formulae give different estimates for population size, give 2 further conclusions about the difference in population estimates given by the Lincoln and Chapman formulae, right? So we need to actually look at these numbers and interpret them. So here, the Lincoln estimate for the large heath butterfly is a lot higher than Chapman, and it's also higher here and there. So immediately, it's just saying they give different values. What else can you say? Well, the Lincoln one gives a higher estimate. They haven't told you that in the question. They've just said it's different. You could say what the difference is. So Lincoln gives a higher estimate, right? If you compare here and here, the difference is about 300, right? Whereas here, the difference is about 10. The difference here is about 300, the difference is about 10, right? If I scale this up, let's say we had 3,000 to 2,000, right? Estimates, to make them so we can have the same number of figures. That estimate would, in theory, should be 1,000. Can you see how that difference is much fatter versus here? So it seems like having a smaller number has a proportionally larger difference in the population. Also, you can kind of see it here, 1 being remarked and stuff, and then slapping into the formula, 5 and 6 versus 4 out of 77 and 73. So the idea here is that you have if you have a smaller population size, you then, for some reason, tend to have a greater difference between the estimates between the two. There you go. Alternatively, you could work out the percentage of the difference of the original things and also do it that way. But that's a quick shortcut way. So the peat bog habitat has been damaged by peat extraction and management of the neighboring farmland. Ecologists decided to treat the peat bog in the following way. So a buffer region was created between the peat bog and the neighboring farmland. No visitors are allowed. Ditches were blocked to raise water levels. A peat extraction, tree planting, and the use of fertilizers were banned. A student suggested that this is an example of preservation. So evaluate their conclusion. So when it says evaluate, what's supporting it, what's contradicting it, right? So if we're looking at supportive statements, remember that preservation is where you are doing nothing. You are just leaving it the way it is. You're not removing anything. You're not putting back in anything, and you're stopping humans from doing anything, right? And you're just leaving it to do its natural thing, right? Whereas conservation is different, because conservation is where you're actively doing things to manage that. So you can see there's a mixture of the two going on here, because there are not allowing humans to do anything, but there's also still human interference, like this step here. You can't just say no human interference, because immediately there's that, right? And also we've created a whole region here. So the idea is we need to pick out what matches preservation and what doesn't. So supportive statement here is that no, for example, no visitors are allowed. That's stopping human activities, right? So we're stopping human interference here. But what's contradicting is that the habitat is being actively managed, e.g. the creating a buffer region and blocking ditches. There you go. You have to just be careful to read the question. Don't unload everything about preservation and just absolutely nothing when it's ever an evaluate question. What's supportive? What's contradicting? And there you go. And whether it's an experiment as well, validity things. So conservation and biodiversity, we have international and national agreements to help with things. So the Environmental Countryside Stewardship Scheme is a national thing the UK has. And basically it involves saying, look, bro, if you do X and Y, to help with, conservation. We'll give you like nice incentives. We'll pay you. We'll do something nice, right? That's a UK government specific thing. Whereas the other ones, so if you look at the Convention on International Trade in Endangered Species, the whole point of that is that it's an international thing where they all came and said, look, lad, you're not allowed to kill endangered species, and you're not allowed to trade with their products, like for example, rhino ivory. Okay? The idea is that conserves them species and stops those species, and stops them from getting endangered, and that's an internationally agreed thing, right? The Rio Convention on Biological Diversity involves international strategies that like countries came along and said, look, here's what we're going to do. We're going to try and make things and come up with guidelines and rules and things like that and laws to make sure that animal and plant resources are used sustainably, meaning we don't run out of them for future generations. And that's part of international law. And it also gives guidance and guidelines to governments as well on what they can do to conserve biodiversity. So in summary, Environment Countryside Stewardship Scheme is our UK specific thing where it's like, all right, lads, if you do X and Y to conserve these, we'll give you money and we'll pay you and give you nice treats and presents. The Rio Convention on Biological Diversity is an international thing where we come along and say, look, let's make these uses of animal and plant products more sustainable. However, they also still do produce guidance for governments for like the UK to use. And then the other one, which is the Convention on International Trade in Endangered Species, stops things like killing and hunting and selling products from endangered animals like rhino ivory. And that's an international thing. So then here, that one is a local thing where we're paying them for incentives and stuff like that. This is an international thing. That's an international thing. So there you go. The oxygen dissociation curves for adult and fetal hemoglobin are shown here. One real quick thing about affinity, right, is I don't like sitting here and memorizing, oh, this is why it goes left, this is why it goes right, and then some weird acronym that low-key just doesn't make sense and barely works. You can just draw, if you want, a really quick sketch. If I draw a super quick sketch, if you just have a vague idea of how these curves work, then it gets quite a lot easier, right? Here, low PO2, it's hard for the hemoglobin to bind to oxygen because there's barely any oxygen for it to. Once the first couple bind, right, it starts changing shape, the hemoglobin, to make it easier for the rest to bind, so you get that short uptake. And then here, because we're in the lungs and the partial pressure of oxygen is really high, there's a very few binding spots left for the oxygen to bind on hemoglobin, so that's why it levels off. If you think about it, high PO2 is going to be your lungs, low PO2 is going to be the rest of the body. So if you follow the curve going down, right, the saturation is dropping, so it's dissociating, it's unloading, right? If it's got a higher affinity means how well you're holding on to something. So if I have a really high affinity, I want to hold on to it for longer before I drop, bang, like that. So the curve shifts that way. Whereas if I have a low affinity, I want to give it up more readily. So rather than waiting to drop here in this PO2, I'll just drop it earlier. That's how I memorize which way the curves go, because I just can't be bothered learning really weird acronyms. So here, this is where it's dropping for whatever, that's adult, right? And then this dotted one is fetal. And if I have a look, because it's holding onto it for longer before it drops, that means it's got a higher affinity. Done. Oh, and here you go. It says outline why it's more important for fetal hemoglobin to have a higher affinity than adult. So compare what's going on. If you have a higher affinity, it means you want to hold on to oxygen for longer. We get our oxygen, we adults, we say, from our lungs, which is where our PO2 is really high. The fetus is getting it from the placenta, which is not the lungs. You need to think, why on earth would a fetus want to grab onto as much oxygen as possible when it's getting the oxygen from the placenta? The only fundamental difference is lungs versus placenta. The idea is that there must be low PO2 in the placenta compared to the lungs. So there's a lower PO2 in placenta versus the adult lungs. Therefore, a higher affinity means that there's more O2 transfer from the placenta to the fetus because it wants to grab it more, right? If that's the case, then there's enough O2 for aerobic respiration. Generally speaking, if you're talking about amounts of oxygen and things like that, right? It's a pretty good thing to just talk about aerobic respiration and ATP because that's kind of what we need it for, and ATP production. There you go. Myoglobin's found in muscles. Oxygen binds to myoglobin. A student described the oxygen dissociation curve as follows. So when oxygen first becomes available, myoglobin saturation increases at a constant rate of 8% per mmHg of oxygen. When there is a slightly higher partial pressure of oxygen, the rate of oxygen binding slows gradually until the myoglobin is 100% saturated. The partial pressure at which this reaches 100% saturation is the partial pressure at which the adult hemoglobin is about 80. Sketch an appropriate dissociation curve based on that description, so we need to use those numbers. So here, it says here, when the first oxygen is available, it starts increasing at a constant rate at 8 per minute. So there's not really any help about where it starts, but we can have a look at some other bits and bobs, right? It says here, when there's a slight one, blah, 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 it slows gradually until it's 100% saturated. And where it becomes 100% saturation is where the adult is 80. So we'll go to where the adult one is, find 80. So the adult one being 80 is here, right? So this mysterious myoglobin is going to have saturated way higher up here immediately, right? Cool. But then it's also told us that when it's hitting that 100, it's also, well, where is it? So it increases at a constant rate. So the sharp straight line is a rate of 8%, sorry, per millimeter Hg of oxygen. So if we think about it, this adult one hits its like, full 80, sorry, it's 80% saturation at a value of about what, 50, that's 50, 52, 53-ish, 52, right? So 52 mmHg, right? So in that case, this myoglobin will start leveling out at that 52 as well, right? It hits that 100. So prior to that, it will have been this, and then it would have had like that sharp straight liney bit there. So then if it's going at 8, right? Because that's what they've told you, it's 8% per mmHg of oxygen, then if I draw that out, then we just have to keep doing 1 mmHg and a rate of 8, if that makes sense. So if I'm here now, which is about what, so if I look at where this straight line's about to start, that's at what, like, okay, I'm being very generous with that line, but it's about 30, right? 2, 4, 6, 8, 10, right? 30. It's the question's told you that each and every single millimeter of oxygen, mmHg of oxygen, you go at a rate of 8%. So if I'm at 80, then each square means I'm dropping by 2, right? So just do a quick bunch of plots. Each square, remember, is like 2 mmHgs. So I'm going to drop by 2 lots of 8 every single square. So that means I'm going to drop by 16 for here from this square to the next, which means I'm going to be at 64. So then for my next one, wherever that may be, so we're moving 1 square to the side, it's going to be where 64 is, which is about there. Then the next fall is going to be 64 minus 2 lots of that 8 again, because again, we're moving 1 across 1 square, right? Which is now at 48. So then I can just trace down to where 48 is for the next square, which is about there, right? And then the next drop is going to be 48 minus 2 lots of that 8, which is going to be 32. So my next square is going to be wherever 32 is, which is about there, right? For the next square to the side. So about here, right? Can you see how now we have that sharp decrease there? You just draw your sharp line and level it off like that. There you go. So the idea here is you're just sort of drawing the bits based on the information they gave you, and that makes a lot more sense given the question context. So hemocyanin is oxygen-binding pigment that's found in many invertebrate animals, including lobsters, and here's the oxygen dissociation curve for hemoglobin and hemocyanin. If you have a look, the hemocyanin's holding onto the oxygen way longer, and then bang, it drops it. So that means the affinity is higher. It says, what can you conclude about the habitat of a lobster? If this is holding on to oxygen for more, it means that it must be in an environment with lower oxygen immediately. Habitat, lower O2. That's it. Because why on earth would it want to hold on to more? Like, lobster's not really doing very much else. There you go. When old red blood cells are broken down, each heme group is converted to a molecule called bilirubin. Bilirubin passes through the digestive system and it gives feces their characteristic color. Explain why bilirubin production and processing is an example of excretion. So it's a metabolic waste. I mean, you're pooping it out. So that's immediately one of them. So that, because it's being pooped out, you can insinuate that it's a waste product. And that's also something you need to know anyway, right? So it's a bilirubin is a waste product made from RBC breakdown, right? The whole definition of excretion is the removal of waste products from the body. So if it's a waste product, we need to talk about how it's being removed from the body. That's the whole thing there. No need to, exaggerate this or like over-explain this. It's just you've got to turn it into context and put it into this context here. So a lot of people, you knew what excretion was. They just couldn't put it into words here. All it's saying is excretion is the removal of waste products from our body. So you need to say here, whatever the waste product is, where it's from, and then it being removed from the body. There you go. Anyway, I hope that helps. Please like, share, and subscribe. You know the drill, and thank you very much.